In the last session, we had completed discussion about the entire reproductive system of both male and female in case of human being. Next one, we will pass it over to the concept of a gametogenesis. In the first chapter, reproduction and organism chapter, we come across a word called gametogenesis. Genesis stand for formation or synthesis. Gametes, nothing but the haploid cell which are being used in the case of process called fertilization. So, gametogenesis can be defined as the process of formation of haploid gametes. Formation of a haploid gametes, that event we are going to call it as gametogenesis. In case of human being, as we all know, human being is a unisexual individual, male or female sex organs will be there in the case of an individual. That is why there is two kinds of a gametogenesis process will be there. One is a spermatogenesis. Spermatogenesis. Second one is a oogenesis. So, two kinds of gametogenesis process or gamete formations will be there with the reference to human being. One is spermatogenesis, second one is the oogenesis. Now, one by one we take the concept into consideration for the discussion. First one, event is spermatogenesis. Now, how I can define the word spermatogenesis? It is the process of formation of haploid sperms. In the testis, we can consider as a spermatogenesis. So, it is the formation of a haploid, single set of chromosomes will be there. That is why we use the word haploid. Sperms, alternate word for the sperm is spermatozoa in testis. Probable one mark question connected to the term called spermatogenesis is location, where actually the event is going to take place. In the last session, I have told you, or in a previous slide, I had told classroom discussion, I have told you, there will be an testicular lobules will be there. Testis will have the 250 conical shaped compartment we call testicular lobules. In the testicular lobule, there will be a seminiferous tubules. In the seminiferous tubule, on the inner lining of a seminiferous tubule, if I taken, it will have the cells called spermatogonial cells will be there, like this spermatogonial cell, plus there will be a pyramid shaped structure will be there called Sertoli cell. These spermatogonial cell has the ability to undergo meiosis and produce the sperms. Then the location of a spermatogenesis or a site also I can use. Location or a site of a spermatogenesis is in the seminiferous tubules. In the seminiferous tubules of a testis. Next point, probable another one mark question connected to the spermatogenesis is time. When actually spermatogenesis will going to start. This statement I am taking into consideration because oogenesis also concept is there in our syllabus. They will say oogenesis or egg formation of female will going to start during the fetal stage itself. Then in the embryonic stage itself, what you call the egg formations will going to start. In case of a male, spermatogenesis or sperm formation starts during puberty. During puberty, when the boy had reached the age of 12 to 16, at that time the sperm formation starts in the case seminiferous tubule, obviously under the influence of hormones produced by the anterior lobe of pituitary. Steps of spermatogenesis. There will be two main steps will be the first event is spermatogenesis. First event is spermatogenesis. Second event is spermiogenesis. So, first one is a spermatogenesis, second one is a spermiogenesis. If not, we can use alternate word is spermatogenesis. is another terminology for spermiogenesis. We will going to discuss these things in detail. This concept will going to come for 5 mark in the board examination. Very popular concept in the case of a board examination. So, first step comes in the case of spermatogenesis is spermatogenesis. Spermatogenesis. Genesis already told you formation of a spermatid. What is the characteristic of spermatid? Spermatid usually spherical in shape and they are non-motile. Then spermatogenesis is the formation of a non-motile spermatid or spermatids. We are going to call it as spermatogenesis. During the spermatogenesis, what is going to happen? In the seminiferous tubule, if I taken, this is the inner lining of the seminiferous tubule, and here there will be an interstitial cells also going to come. There will be basal lamina. We know it, epithelium. When we say the cells will be rested on the membrane called the basal membrane. Now, on the basal membrane, there will be spherical shaped structures will be there. Like this, 4 to 8 cells will be there, which are arranged one above the other. Like this, cells will going to come. These cells are known as spermatogonial cells. And in between, there will be a pyramid shaped structure or pyramid shaped cell will be there, that we call it as a Sertoli cell. This is the sectional view of a seminiferous tubule. So, this is the Sertoli cell. In the previous classroom discussion already I told you, Sertoli cell I can use, or sustentacular cell I can call, or nurse cell also acceptable word. Here the lower cell is the spermatogonial cell. Spermatogonial cell. This cell will be diploid, this cell also will be diploid in nature. Now, what happened? This spermatogonial cell, which is present in the case of seminiferous tubule, will undergo repeated mitotic division. Diagrammatic representation or schematic representation, 2n cell is spermatogonial cell, which will have 46 chromosome. 2n is equal to 46, that is 44 will be autosome, somatic chromosome, plus X and Y chromosome, because we are discussing related to human being male. Male do not have a 2X chromosome, male will have 1 X and 1 Y chromosome. Now, this spermatogonial cell undergo repeated mitotic division, undergo mitosis. We know it is an equational division. Mitosis division is also known as equational division. We will going to get more number of cells. Again, these cells will undergo repeated mitotic division. The division again is mitosis. Now, there will be increase in the number of spermatogonial cells. All these cells will have diploid set of chromosomes. So, this is the first event. Here, what are the spermatogonial cells will be there? These cells undergo repeated mitotic division and they are going to increase their number. They are going to increase their number. Now, most of the cells will be, or I can say generalize the point, type A and type B cells. Type A and type B cells will be there. Type A cell, assume theoretically these cells will remain as the spermatogonial cells only. Remaining B cell, what will be there, will become primary spermatocyte. There is slight increase in the case of size will be there. Now, this cell will enter into phase of a growth. Now, it will going to behave as a what you call the primary spermatocyte. primary spermatocyte like a spermatogonial cell also diploid in nature. 2n is equal to 46. Now this primary spermatocyte will undergo meiotic division, that to meiosis 1. Meiosis 1. When they undergo meiosis 1, it is a reduction division. First PUC cell reproduction chapter, we will going to get the two cells which are haploid in nature. Like this, two haploid cells we are going to get it. These haploid cell we are going to call it as secondary spermatocyte. We call it a secondary spermatocyte, which are haploid in nature, formed by the meiosis 1. Each secondary spermatocyte will undergo meiosis 2. Undergo meiosis 2 and will produce haploid spherical shaped non-motile structures. Like this, there will be an haploid spherical shaped non-motile structure. These structures we are going to call it as spermatids. We are going to call it as a spermatids. So, one primary spermatocyte will produce 4 spermatids which are haploid in nature. So, this event is called spermatogenesis, meaning is formation of a spermatid from the cell called spermatogonial cell. So, whether the spermatogonial cell directly going to give rise to spermatid by undergoing only meiosis? No. They will undergo repeated mitotic division and going to increase their number. Some of the cell remain as spermatogonial cell to produce the sperms in the future. Assume if this is the spermatogonial cell directly has undergone the meiosis, and that means this spermatogonial cell will directly become 4 sperm. Whether in the future spermatogonial cells will be available to produce what you call the sperms? No. That is why there will be mitotic divisions that takes place in the case of beginning. Some of the cell remain as spermatogonial cell, some of them will going to enlarge in a size and become primary spermatocyte, which will be diploid like the spermatogonial cell. Undergo meiosis 1 and each primary spermatocyte going to give rise to 2 haploid secondary spermatocyte of equal in size. Each secondary spermatocyte undergo meiosis 2 going to give rise to 4 haploid spermatids. This much event is called spermatogenesis. Now, in the case of the diagram, if I taken this is the spermatogonial cell. Next one more cell will going to come. Now, I can call this one as a primary spermatocyte. Primary spermatocyte. Above that, there will be a cell will be there. I can call it as secondary spermatocyte. From the secondary spermatocyte, we are going to get the haploid cell. That cell is nothing but the spermatids. Spermatids. So, these are the points related to the sectional view of what you call the testis, and this is the process of formation of a spermatid. Next event. In the spermatogenesis, I told you one first event is the spermatogenesis. It will be followed by the next event. The next event is spermiogenesis. Next event is spermiogenesis. Spermiogenesis, also known as spermatogenesis. Spermatogenesis. By spermatogenesis, we will going to get the spherical, non-motile, haploid spermatids. That spermatid will change us into motile, flagellated sperm. That event is spermiogenesis. During the process of spermiogenesis, there will not be any mitotic division nor meiotic division. Divisions will not going to take place. Instead, there is only structural changes will going to take place in the case of a spermatid. That is why for the spermiogenesis, I will say it is the morphological. What is the meaning of the word morphological? First PUC syllabus, structural. Morphological conversion or changes, conversion of a spermatids What is the care feature spermatid? Which is non-motile. Why they are non-motile? Because they do not have a locomotory organelles. Remember, sperm have a locomotory organelle. First PUC concept, flagella, locomotion movement chapter if you recall. So, in the spermatid they are non-motile because they do not have a locomotory organelle. 2. Sperm which will be motile in nature. This event we are going to call it as spermiogenesis. Now, how the spermiogenesis will going to take place? During the spermiogenesis, what the spermatid has been formed from secondary spermatocyte will be attached to the structure called Sertoli cell. Then I can say spermiogenesis site is on Sertoli cell. On the cell is nothing but the Sertoli cells, where when the spermatid, as already told you, spherical in shape, they have a lot of cytoplasm will be there. Flagella will not be there. Golgi complex is not developed into acrosome. These are all the structure has to develop now. That is, in the case of the spermiogenesis, there will be a formation of acrosomes will be there. Formation of acrosome. Acrosomes will be formed from a Golgi complex. Golgi complexes will going to produce us the acrosome, which is necessary for the fertilization event. Second one is condensation of nucleus. The haploid nucleus of a spermatid should undergo condensation. Third one, there will be an elimination of excessive cytoplasm. Elimination of excessive cytoplasm that will takes place from the spermatid. Next one, there will be an formation of axonema. First PUC point, cell the unit of life chapter. We know in a eukaryotic cell there is a locomotory organelles. Locomotory organelles are flagella and cilia. In a eukaryotic flagella and cilia, the center core is axonema, and we know it is made up of microtubules. 9+2 arrangement of what you call the microtubules will be there. So, there is a microtubules assembly should takes place. That center core of a flagella or cilia we are going to call axonema. Here the flagella will be there. So, there is a formation of a axonema or axonemal filament. Fifth point, there will be an assembly of a centriole. Assembly of a centriole will going to take place. So, like this, when there is a morphological changes, structural changes will going to take place. Spherical shaped, already told you, spermatid will be spherical shape. We will going to change us to the tadpole shape. Tadpole shape, spermatozoa. That event we going to call it as spermiogenesis or spermatiliosis. Spermatiliosis. Now, so two main events. Two main events will be there in the case of a spermatogenesis, sperm formation. First one is spermatidogenesis, second one spermiogenesis. During the spermiogenesis, there is a second step of a spermatogenesis process, head of the sperm, because already told you sperm, matured sperm of a human being will be tadpole shape. Tadpole shape. Now, what is meant by tadpole shape? A structure which have a head and a tail, which will have a two-part head and tail. Such structure we are going to call it as what we call the tadpole shape. That terminology you have studied in the first PUC in the chapter biological classification. In a biological classification, the last page they had given the diagram of a T4 bacteriophage or bacteriophage, which will have a head part as well as a tail part. That we call it a tadpole shape. So, similarly, in the case of a sperm, of human sperm when we taken, it will be tadpole shape because our two main part will be the head as well as the tail part will be there. Now, once the sperm has been formed, it will have a part called a head. The head portion of the sperm will be embedded in the structure called Sertoli cell. After that, there is an event will be there called spermiation. One of the very popular 1 mark question in the examination. Word is called spermiation. So, what is spermiation? See here, assume this is the seminiferous tubule which will have a cell called Sertoli cell, and here there is an attachment of a spermatozoa will be there. Spermatid will be spherical shape. When they are attached to the Sertoli cell, they changes into tadpole shape. This is the head and this is the tail part. This head part will be embedded inside the Sertoli cell, and after that sperm will going to come out. That event we are going to call spermiogenesis. So, what is it? It is the release of, first let me take the sentence according to NCERT. It is the release of sperms into lumen of seminiferous tubules. Lumen of seminiferous tubules, we are going to call it as spermiation. Or another way, the sentence which got appeared in the case of NEET examination is release of the matured sperm from the cell. Which cell? Sertoli cell, we are going to call it as spermiation. So, it is the release of a sperm from Sertoli cell. Sertoli cell. So, both these sentences are applicable for the concept called spermiation. So, spermiation is not the part of a spermatogenesis. Spermatogenesis are only 2 steps. First one is spermatogenesis, second one is nothing but the spermiogenesis. So, these are the points related to the concept. What the schematic representation have just shown, that is going to come for the board examination also for 5 mark. The steps involved in the process of what you call the spermatogenesis. Next one, there will be an hormone response will be there. Hormones regulating male reproductive system. Hormones will be there which is going to regulate the male reproductive system. In the first PUC chapter chemical coordination, we have studied hypothalamus will be there. Hypothalamus is a one gland which is going to secrete both the releasing hormone as well as inhibiting hormone. Example for releasing hormone in the first PUC we have studied is gonadotropin releasing hormone. Then we have studied about the antelope pituitary. Antelope pituitary will have a tropins. Tropins are, one is FSH, follicle stimulating hormone, and second one is a luteinizing hormone, 2 hormone. When you taken the testis into consideration, we have studied there will be testicular androgen will be there. These are all the names we have studied in the case of first PUC. Same concept will be taken into consideration to explain the hormones regulating the male reproductive system. First one, as we know, there will be a gland called hypothalamus, which is a part of brain. In the case of the brain, there is a diencephalon. Diencephalon will have a part like epithalamus, thalamus and hypothalamus. So, this hypothalamus during puberty, because we are discussing about the male, not in a case of female. So, during puberty, hypothalamus will go to secrete a hormone. Hormone secreted hypothalamus is GnRH. GnRH. GnRH stand for gonadotropin releasing hormone. Gonadotropin releasing hormone. This is a releasing hormone secreted by the hypothalamus. The site of action of hypothalamus is anterior lobe of a pituitary. So, it will going to stimulate the anterior lobe of pituitary. In first PUC, we have studied the releasing hormone produced by the hypothalamus and as well as the inhibiting hormone produced by the hypothalamus come to the anterior lobe of pituitary through hypophyseal portal system. Diagram is given in the case of first PUC related to the hypothalamus pituitary gland structure. There you can see there is a network of capillary One is present in the hypothalamus, another network of capillaries present in the adenohypophysis. Adenohypophysis is another name for anterior lobe of pituitary. So, anterior lobe pituitary will be stimulated by GnRH, and GnRH reaches anterior lobe pituitary through hypophyseal portal system. Now, the anterior lobe pituitary will going to secrete two hormones. One is FSH, that is nothing but the follicle stimulating hormone. Second one is LH, stand for luteinizing hormone. So, two hormone FSH and LH will be secreted. What is the cat feature of FSH and LH? Both are gonadotropins. Gonadotropins. Gonadotropins mean tropic hormone. Hormone produced by the anterior lobe of pituitary stimulate the other endocrine gland. That endocrine gland in this part is nothing but the testis as well ovary. Testis and ovary have an endocrine property also as well as gametogenic property also will be there. So, gonadotropin means tropic hormone produced by the anterior lobe pituitary act on gonads. They are FSH and LH. FSH will go to act on the cell which is present in the seminiferous tubule, that is nothing but the Sertoli cell. They act on the cell called a Sertoli cell. Luteinizing hormone LH will be there, they will go to act on the Leydig cells. They act on Leydig cells. So, Leydig cells I can use, or I can use the word interstitial cells of the testis also acceptable. Leydig cells now produce one hormone called testosterone. Produce a hormone testosterone. Since LH luteinizing hormone act on the Leydig cell and make them to produce what called testosterone, LH also known as interstitial cells of interstitial cell stimulating hormone. Stimulating hormone, abbreviation ICSH. So, there will be interstitial cells will be there on which the LH will going to do the work, that is why interstitial stimulating hormone. When a FSH act on the Sertoli cell, now Sertoli cell produces two compounds. This word is not given in the case of the NCERT textbook. Instead, what they mentioned is Sertoli cell produces some protein factors that produce some factors. That protein factor is nothing but the ABP, produce a chemical compound called ABP. So, what is ABP? ABP full form is androgen binding protein. Androgen binding protein will be there. That will be produced by the Sertoli cell. Then what is the function of a androgen binding protein? Androgen binding protein will go to concentrate the testosterone in the case of Sertoli cell. ABP concentrate the testosterone in the case of Sertoli cell. Because of that, there will be an event will be there called spermatogenesis. Then here I can write it will helps in the case of spermatogenesis. Spermatogenesis will be there. Sertoli cell also produce one inhibiting factor called inhibin. Produces a hormone called inhibin. When the rate of spermatogenesis will be high, more number of sperms will be produced per unit of time, then this inhibin will go to inhibit the anterior lobe of pituitary by negative feedback mechanism. Negative feedback mechanism means a part of a homeostasis mechanism where original stimulus is been altered. What is the original stimulus? Original stimulus is producing more number of sperm. Now, that producing more number of sperm has to be stopped. That is why we call it the negative feedback mechanism. So, inhibin act on the anterior lobe of pituitary and inhibit the secretion of a follicle stimulating hormone. When a FSH is not secreted, it cannot act on the Sertoli cell. Sertoli cell cannot produce the ABP, concentration of a testosterone is not possible. So, concentration of a testosterone is not possible, automatically rate of a spermatogenesis will going to decreases. Similarly, when there is a more amount of testosterone will be produced, they again have an inhibitory effect will be there, negative feedback mechanism will be there. it will going to act on the anterior lobe of pituitary as well as the hypothalamus and inhibit the secretion of a GnRH as well as FSH and LH. So, this is nothing but the schematic representation of the role of various kinds of a hormone on a male reproductive system. So, here we can write, here also we can write spermatogenesis, or here under this one also we can write the event called spermatogenesis, because testosterone required for what called spermatogenesis. Broad outline of the concept: during puberty, hypothalamus secrete GnRH. GnRH act on the anterior lobe of pituitary and secrete FSH and LH. FSH act on the serotolin cell and produce inhibin and androgen binding protein, which helps in the case of spermatogenesis. How ABP helps in spermatogenesis? ABP concentrates the testosterone in a serotolin cell and increase the rate of what you call this spermatogenesis. Luteinizing hormone will going to act on the leading cells. The leading cells or interstitial cells of the testes secrete the hormone called testicular androgen, mainly testosterone. That is testosterone necessary for spermatogenesis. Now, regulation process by negative feedback mechanism, where inhibit the anterior lobe pituitary and inhibit the secretion of a follicle stimulating hormone. Testosterone inhibit the hypothalamus as well as anterior lobe of pituitary. So, this is nothing but the hormonal control of a male reproductive system. Next one, we will pass it over to another probable 5-mark question in the case of board examination. It is nothing but the structure of a human sperm. Structure of a human sperm will be there. Probable 5-mark question in the case of board examination. First one is about the shape. Already I have told you the shape of a human sperm is a tadpole. Tadpole shape will be there because it will have a two main part, head as well as the tail part will be there. That is why it is called tadpole shape. Size, if I taken around 60 micrometer in a length. 60 micrometer in a case of a length. That is about the size of the human sperm. Now, parts, if I taken according to NCERT concept is considered, it will have first part called head, second part is a small neck will be there, next one is a middle piece, and last one is a tail. So, like this there are 4 parts will be there with reference to the structure of a human sperm. One by one we take the concept. First one, the anterior part of the human sperm is nothing but the head. Anterior part means part of the body take a lead at a time of locomotion, we are going to call it as the anterior part. Human sperm head, if I taken, it will be pear shaped. Remember, it will be pear shaped. Like this structure will be there. And the entire structure of a human sperm, head, neck, middle piece and even tail part will be covered by plasma membrane. Covered by plasma membrane or also known as a cell membrane. So, that is why in the case of head part also the outermost covering is nothing but the plasma membrane. Inside the head of the sperm, it will have one nucleus will be there. So, this is the nucleus which is haploid in nature, single set of chromosomes will be there and it will have a 23 chromosomes, 22 autosome, 1 X or 22 autosome, 1 Y chromosomes will be there. That is about the nucleus. On the anterior part or above the nucleus, there will be an cell organelle will be there. which is formed from the acrosome like this. This structure we are going to call it as acrosome, generalized giant lysosome or acrosome. This acrosome contain hydrolytic enzyme. So, anterior to the nucleus there is a acrosome. This acrosome has a hydrolytic enzymes. Example one is acrosome, second one hyaluronidase, etc. Like this there will be an hydrolytic enzymes will be there in a case of acrosome. So, is there any function will be there of a acrosome? Because since as I told you they contain hydrolytic enzyme, they helps in the case of a fertilization, helps in the event called a fertilization. Fusion of the gametes, fusion of the sperm and egg is possible because of the presence of a structure called a acrosome. It will be followed by the very small part will be that small part we are going to call it as neck. This is the neck of the human sperm. This much portion is of a head. In the neck region, it will be around 0.3 micrometer in length, comes about 0.3 micrometer. And here we come across two centrioles will be there. One centriole present posterior to the nucleus or present below the nucleus, that we call it as proximal centriole. We call it as a proximal centriole. One more centriole lie perpendicular to the proximal centriole. This is a 2D structure. 3D structure if it is, then we can able to understand it in better way. In a 2D structure I can't show. So, proximal centriole and distal centriole will be there which are perpendicular to each other. So, beneath the proximal centriole I can write there will be a distal centriole. Distal centriole. Only the flagellated human cell is a human sperm. No other cells are there in a human body which are flagellated. Most of them are ciliated or maybe amoeboid. Ciliated will be found in the trachea, or in the case of fallopian tube we come across the ciliated cells. Some example. Pseudopodia, locomotor organelles will be there in the WBC. In the macrophages we come across these kind of structures. Flagellated cell is only human sperm. So, what is the center of the human flagella has? Center of the human flagella has an axonema. That axonema arises from the distal centriole. Remember. So, axonema. Axonema is also known as the axial filament. Correct now? First PUC syllabus, axial filament, and we know this axial filament made up of a microtubule which are in 9+2 fashion. Duplet of 9+2 fashion microtubules will be there, which arises from distal centriole. Distal centriole and run along the length of a sperm. Third structure of a human sperm is a middle piece. Next one is a middle piece. Like this, there will be a part will be there. This part we are going to call it as middle piece. In the middle piece, in the center, it will have a axonema. It will have a axonema. Plus, it will have a 9 peripheral fiber also will be there, and it will surrounded by the mitochondrial sheath like this. This is a 2D structure. Like this, there will be mitochondrial sheath will be there. Unique feature of a middle piece. So, it will have a axonema which will have a 9+2 microtubules will be there. Next one, it will have a 9 accessory fiber also will be there. And it will be important point about the middle piece is having the mitochondrial sheath. It has a mitochondrial sheath which are arranged spirally around the axonema. Usually we use the word mitochondrial sheath. If not, we can use alternate word Nebenkern. Also acceptable is another terminology for mitochondrial sheath. We know mitochondria role. Mitochondria will generate ATP by oxidative phosphorylation. So, this sperm will show what we call the rigorous movement. For the movement, it required energy. That energy will be provided by the mitochondrial sheath. So, mitochondrial sheath provide energy for the movement of sperm. For the movement of a sperm, energy is required, that will be provided by the mitochondrial sheath. That is why in the board examination we can ask the questions like this. Middle piece of human sperm is considered as a engine room of sperm. Engine room of the sperm, because engine will going to generate the energy required for what we call the movement. Similarly, mitochondrial sheath will go to generate ATP required for the movement of the sperm. So, these are all the points related to the third part of the human sperm, that is nothing but the middle piece. After that, there will be a part will be there called tail. If I elaborate the point, tail will have a principal piece as well as end piece, like the two parts will be there. But according to NCERT, it has given as it will have a tail part. So, in the case of tail part, it will have an axonema center like this, and that axonema on the external side will be covered by the plasma membrane. Like this, there will be plasma membrane will be there, but in the case of a tail, mitochondrial sheath will be absent. So, this is nothing but the tail portion. Tail. So, in the case of a tail, it will have an axonema, or I can consider there will be a microtubule arranged in a plus fashion of a 9+2 fashion. Second one, no mitochondrial sheath. Mitochondrial sheath will be absent. If you want to elaborate the point, then we can add, not required for the board exams as such consider. It will have an axonema. There will be 9 axillary fiber, then there is a fibrous sheath terminology will be there connected to the tail part. And after that, there will be and last part will be called end piece. Together we consider as what we call the tail. So, these are the various parts connected to what we call the human sperm. So, human sperm will have a first part called head, next one neck, middle piece and tail. Head have a two important cell organelle. One is acrosome located anterior to the nucleus with a hydrolytic enzyme like hyaluronidase. Next one acrosome, like that enzymes will be there which are required for penetration of a sperm into the egg during fertilization. Then below the acrosome there is a nucleus which is haploid in nature and have a 23 paternal chromosome. 23 paternal chromosome. Paternal mean father. So, it is because sperm will be formed in the case of a male. So, it will have a 23 paternal chromosomes. Next one, it will have a small neck around 0.3 micrometer in length and have a two centriole, proximal centriole and distal centriole. Proximal centriole is present posterior to the nucleus, which will going to enter into the egg during fertilization and helps in the cleavage division. Helps in the cleavage divisions of a egg after fertilization. One more is a distal centriole which is located perpendicular to the proximal centriole from which axonema arises which run along the length of the sperm. Middle piece has a axonema followed by the mitochondrial sheath. That mitochondrial sheath we call Nebenkern arranged spirally that generate energy required for the movement of the sperm. followed by the tail, which do not have a mitochondrial sheath, which is the longest part of the human sperm we can consider. In the last session, I have told you semen will be there. Semen. Now, what is semen? Seminal plasma. What is seminal plasma? Fluid secreted by the accessory gland of a male reproductive system. Fluid secreted by the accessory glands of a male reproductive system, we are going to call it as what we call the seminal plasma. With the sperm, we are going to call it as a semen. So, semen is the seminal plasma, and sperm we are going to call it as a semen. Whenever the sexual arousal will be there or during sexual intercourse will be there, male is going to eject 3 to 5 ml of a semen. This is the volume of the semen will be ejaculated during an orgasm or during the sexual intercourse. Now, that 3 to 5 ml of the semen contains how many sperms will be there? So, it contains 200 to 300 million sperms. 200 to 300 million sperms will be there. So, this much of a sperm will be there in 3 to 5 ml of the semen, not 1 to 2 ml. It is 3 to 5 ml of the semen contains 200 to 300 million sperms. In the case of the, if I say person is fertile, mean to say capable to produce what you call the children, fertility concept that comes in a case next chapter, that is in human reproductive health. At that time we will let you know what is fertility and infertility concept. If I consider the male is a fertile male, when actually we will consider the fertile individual as a fertile male, the known volume of the semen person what has ejaculated should have 60% of a sperm, should have a 60% of sperm with the normal size and shape. Should have a normal size and shape. Then we consider a person will be considered as a fertile male. Then minimum 40% of the sperm should have vigorous motility. Vigorous motility. If these things are there, then we consider the individual fertile male. This according to NCERT is considered. So, we have completed discussion about the male reproductive system plus spermatogenesis, sperm and hormone regulation of what you call the spermatogenesis.